Hello and welcome back to my channel. Hope you all are doing well. So as we have been discussing about the series, that is translation series. In the first part of the translation we have discussed about how the protein synthesis is taking place in the prokaryotes. In this part of the video we will be discussing about how the translation process is taking place in the eukaryote. So in the general process of transcript, that is translation, in both the prokaryote and eukaryotes are more or less same. Only difference is the factors. And one more thing difference is the tRNA. If you remember, in case of prokaryotes, the tRNA were first amino acid which is brought by the tRNA was F methionine, okay, or you can say formalin methionine. But in case of eukaryote it will be only methionine, the first amino acid will be. This is the difference, but the general process like the initiation, elongation and termination process will be same, but the factors like the initiation factors, elongation factors as well as the termination factors will be different. And one more major difference was we will be discussing in this part of the video. So let's begin. This I have already discussed, that how the difference between the prokaryotic as well as eukaryotic is seen. In prokaryotic, both the translation and transcription process takes place simultaneously, whereas in case of eukaryotes, first what happens, the transcription is done in the nucleus, then this mRNA which is formed, that is after processing and all. We have already discussed the processing process, that is capping, polyadenylation and the splicing process. After all of this is done, it is transported into the cytoplasm. So you can say that the translation takes place in the cytoplasm in case of the eukaryote, okay. Whereas in case of prokaryote it takes place in cytoplasm only and no nucleus is there in the case of prokaryote. If you know the basic difference between prokaryote and eukaryote, this is all the difference between these two. Next, let's just dive into the topic of the translation. Again I am telling the process is same, that is the mechanism is same, only the factors are different. So this is my handwritten note and I have made it a quick short note. So the process occurs in the cytoplasm as you all know, and I have told you that the process takes place in the cytoplasm. This is important point to be noted down. Next, as you know that in case of the eukaryotes, that is 80S type of ribosome is present. As you know, all of you know, 80S of ribosomes are present. 80S ribosome has two subunits, that is 60S, another one is the 40S. Okay, if you remember that, this is 60S and 40S. In case of prokaryotes, that was 70S, 30S and 50S. This is very important to be remembered. Next, what are the things that are required for the process of translation? Obviously, first is the ribosomes is required. Okay, this is the main thing. Without it, the translation is incomplete, you can see. mRNA is required, charged tRNA is required. What is mean by charged tRNA? tRNA, that is transfer RNA with the amino acid, is known as charged tRNA. Okay. Then initiation factor, IF means initiation factor, EF means elongation factor as well as termination factor. Obviously energy is needed, and peptide transfer is enzyme, that is the transfer of the peptide bond from the P side to the A side to the E side, that is transfer is required. The overall, I have already told you, the overall mechanism is same, only difference is in the factor. The eukaryotic mRNA is monocystonic. This is very important to be noted down. Monocystonic means one protein form one mRNA. This is very important. Whereas in case of prokaryote it is polysystonic. Polysystonic means many proteins will form the one mRNA. In eukaryote, coxsacks sequence is present to which 40S ribosome that is small unit binds. So you know that there are two sequences will be present in the DNA structure, that is the coxsack, another one is known as the clinop. Okay. So the 40s, that is in case of eukaryote, that is the coxarch sequence, a particular sequence will be present to which this 40s, that is the smaller unit, smaller subunit of the ribosome will bind to it. Then next starts the process of initiation. As you have know that the initiation, for the process of initiation, certain factors are required, that is known as the initiation factor. What are these initiation factor? There are 9 initiation factor, like the here one more thing I want to tell you. In case of prokaryote, you were simply mentioning about initiation factor as IF, okay, but in case of eukaryote it is written as small e IF, that is small e denotes the eukaryotes. So here the factors like EFI2, EFI2b, IF3, EF4a, 4b, 4e, 4g and IF5. What are the role of these things I have mentioned in this chart? And this chart or this table is very important, because nearly it is asked in exam from this only. This factors only is asked in exam, nothing else. They will not ask the process. Obviously the process is important, but the factors are important. Which factor is responsible for what process? Okay. As you see that IF2 factor which facilitate the binding of the charged tRNA. Why I am mentioning charged? Because here methionine is the amino acid. So the amino acid methionine with the tRNA is known as charged tRNA will bind to the 40S subunit. So the function of EIF2 is to facilitate the binding of tRNA to the 40S subunit. Next, EF2B and EF3, IF3 will bind to the 40S to prevent 60S binding. As you have seen in case of prokaryote also the similar mechanism was taking. The IF3 factors were binding to that of the subunits of the 40S, that is EPA. Okay, EPA subunits it was binding to it, so that 60S subunit does not bind. When the initiation factor will be ready or complex will be ready, after that only 60S complex will bind, as that of the prokaryote. Next, what happened? EFI4A will remove the secondary structure of the mRNA to promote the 40S binding to the mRNA. As you know, the structure of eukaryotic chromosome is little bit complex type of structure, as you know already. They are very complicated. To remove the secondary structure, to make it simplified, they will remove the secondary structure. Which one? EIF4A, and will promote the 40S binding of the mRNA. Next, EIF4B helps in finding the start codon. What is the start codon? AUG by the 40S tRNA assembly. Okay, so once this tRNA will bind to this AUG codon, that is the initiation codon, then the process will be beginning. EIF4E will binds to the 5' cap region, and 4G will bind to the poly A tail region. Okay. IF5 will bind, release the IF and binding of large subunit and small subunit. So at last which factor will work? This IF5 factor will work. What it will do, after all this the complex is made, it will release all the factor, and finally the 60S subunit will bind and the 80S complex will be ready. So here you can see, yes, this is known as the initiation factor. Okay. So first what happens, 40S 60S was present together, then IF4 1A and 3 will separate it into 2. Okay. Then 60S will be present as it is, then 40S will come, 40S will be attached to many IF2 factor, along with that methionine tRNA. Okay. Then it will form a initiation complex, along with that other factors like the IF2A, 3, 4GB, 4A, etc. all will come. Then they will scan, and then what will happen at the end of the end EIF will bind. It will release all the factors. You can see here all the factors are released here, released out. Okay, once again, so that it can be used again. When another tRNA will come, again it will be used, so it will be released, and then finally what will happen, 60S subunit will come, Here the GTP at GTP is used, energy is used, and then the ATS initiation complex will be formed. Okay. So here GTP is used, that is during the EIF2B factor GTP is used, and here also one GTP is used. So total 2 GTP is used in this process. Here ATP is used also in this factor, binding of this factor, ATP is used, ADP is used. So this is all about the initiation process. Okay, this is the same process, overall process it is written, how the initiation will take place. Next what will happen, that is the AUG and initiator tRNA made present in the P site of the 68. So obviously the first tRNA which will come will bind to the P site only. Okay, this is E site, as you know, this is E site, this is P site, and this is A site. So the first tRNA which will come and it will bind here. Okay, and gradually the process of elongation will begin. As the process of elongation will begin, the second amino acid which will come will occupy the A site, and there will be shift in the codon as there in the prokaryote, as I have already told you. There will be shift in the codon, as a result of which the things, that is the polypeptide which was present on the A site will move to the P side, and whatever will be present in the P side will be moved to the E side, and gradually these things will, again the A side will be blanked, again another amino acid will come and bind. So this process continues till they reach to the stop codon, that is UAG, and once it reach to the stop codon, release factor will come and will release the other, that is the tRNA from the polypeptide, and the polypeptide chain will be released. The overall process is same, only difference is in the case of the factors. Okay. Now let's see the elongation. Next tRNA amino acid with the second will come and bind to the A site, as I have already told you. Peptide bond formation take place with the help of the enzyme peptidyl transferase. This is very important enzyme to be noted down. Translocation of ribosomal complex takes place, as I have told you now. Then second amino acid will come and bind to the A site, and A site is empty again. This process goes on continuous. So what are the elongation factors? That is alpha 1 alpha, that is helps in the amino acid tRNA transfer. 1 beta gamma, release of the EFTU and EF alpha, okay, for releasing of this factor, and EFTU is responsible for translocation. As that of the prokaryote, TU was responsible for translocation. Here these factors like alpha is responsible for transferring of the amino acid, beta gamma will be releasing the alpha group, and T2 will help in the translocation. Translocation means shifting of the ribosome by the one codon. Next comes to the termination process. In eukaryote, single release factor, that is ERF1. In prokaryote, 2 release factors was responsible, but here only 1 releasing factors was responsible to recognize the 3 terminal sequence termination codon, that is UAG, UAA, UGA. Okay. EFR3 stimulate the termination. The polypeptide now will be free from the mRNA and release of the small and large subunit. So once again the same thing will happen. You can see here, this is the mRNA, this is the start codon, this is the stop codon, this is tRNA, this is larger subunit. They will come and form the initiation complex. The initiation will go on, elongation go on, then finally when they come to the stop codon, release factor will come and release this peptide, completed polypeptide. Again the components will be recycled. This is very important for another protein synthesis. So this keeps on continuing, and this is all about the initiation one. So if you are very clear with that of the prokaryotic translation, then you will find this eukaryotic translation very easy. Okay. So I request you all to first watch the first part of the video, because the process I have in the diagram I have detailedly explained what is happening. Here I have mentioned only less diagram, because more or less the process is same, only the factors are involved. So I request you all to first go through the first part of the video, then when you understand the general process, then come to this part of the video and only remember the factors. Next, rest of all, everything is easy. factors are only different, rest all the processes are same. So if you read by comparison between the prokaryote and eukaryote, you can easily remember which all factors are responsible for what. So that's all for today, and we have done with that of the translation, that is protein synthesis part. Next part of the video we will coming with certain other new topic. So I request you all to do share this video among your friends, because these are very basic and important for exam point of view. And also do like, share and comment, and if you find it useful. And till then, take care and have a nice day. Thank you.